The human body, a biological masterpiece, reveals its complexity through genetics. Rare inherited variations, persisting through the ages, offer scientists invaluable windows into evolution and diversity. This exploration examines 10 families with exceptional genetic traits, living case studies that push the boundaries of DNA. We will analyze these remarkable examples, highlighting their impact on life and research. Number 10. The Butin tribe community, Indonesia. Unusual blue eyes. In southeast Sulawesi, Indonesia, the indigenous Butin tribe presents a notable genetic phenomenon: the appearance of strikingly blue eyes within a population that generally possesses dark brown eyes. Unlike the blue pigments associated with European populations, this coloration is suspected to be linked to a rare variation of Waardenburg syndrome, a hereditary disorder affecting pigmentation and, sometimes, hearing. The importance of this observation lies in the community's isolation, allowing researchers to study the persistence of this genetic mutation without significant external genetic influence. The study of the Butin tribe thus contributes to elucidating the mechanisms by which rare mutations are maintained and manifested within specific human populations, enriching our knowledge of genetic diversity and phenotype heritability. Scientists continue to monitor these families to decrypt the expression patterns of these rare pigment genes and understand their evolutionary dynamics over generations by isolating the specific markers associated with this unique phenotypic expression. Their genetic heritage represents a precious resource for island population genomics. Number 9. The Sarker family, Bangladesh. Absence of fingerprints. The Sarker family in Bangladesh is famous for an extreme dermatological peculiarity. Several of its members, across different generations, were born without fingerprints or palmar lines, a condition known as adermatoglyphia. This anomaly results from a mutation in the SMARCAD1 gene, which is essential for skin development. The absence of these characteristic epidermal ridges makes biometric identification via fingerprint scanners extremely difficult, earning the condition the informal nickname immigration delay disease. Research conducted on this family is crucial for mapping the precise role of the genes involved in the morphogenesis of skin patterns. Understanding how a mutation can interrupt the formation of structures so fundamental to identification and fine manipulation offers valuable insights into the early stages of embryonic development and the diversity of human biological traits. This family strikingly illustrates the interconnection between genetics, tissue development, and the societal implications of physical traits, and could unlock new strategies for tissue regeneration. Ongoing study aims to determine the molecular mechanisms regulating the growth of the epidermal ridge. Number 8. Linda Bannon and Timmy, Holt-Oram condition, upper limb development. The story of Linda Bannon and her son Timmy highlights resilience in the face of Holt-Oram syndrome, referred to as Holterosyndactyly in the text, a rare genetic condition that leads to severe hypoplasia or the absence of upper limbs. Beyond the medical dimension, their lives are a testament to human adaptability. They accomplish daily tasks, including complex activities like writing and sports, using only their feet. Although this condition is rare, its study within a family setting allows for the observation of how the brain and body adapt to the absence of primary motor structures. Medical research is interested in neural plasticity and the functional reorganization of the motor system in individuals with congenital limb deficiencies. Their example demonstrates that physical limitations are often overcome by determination and the body's ability to find effective compensatory pathways, providing precious information for rehabilitation and the understanding of neurobiological mechanisms of neuronal and motor adaptation. Brain scans reveal unique patterns of cortical mapping in these determined individuals. Number 7. The Manurang family, Indonesia. Treacher-Collins syndrome, TCS. In the village of Kadongkong, Indonesia, the Manurang family faces the challenges posed by Treacher-Collins syndrome, TCS, a genetic disorder that affects the development of facial bones and tissues. The son, Anuir, presented severe anomalies, notably mandibular underdevelopment, leading to respiratory and feeding difficulties, as well as malformations of the ears and eyes. TCS, which occurs in approximately 1 in 50,000 births, is characterized by hypoplasia of the cheekbones, jaw, and chin, often accompanied by hearing loss due to middle ear anomalies. The study of this family is fundamental for geneticists seeking to target the signaling pathways involved in craniofacial formation. The dedication of the father, Sarif Ali Serja, to raising public awareness of TCS underlines the societal importance of understanding these rare disorders, not only for the complex surgical interventions they require, but also to provide psychological and community support to affected families and to develop targeted gene therapies. Research focuses on the TCOF1 gene, which is essential for the survival of neural crests. Number 6. The Ulas family, Turkey. Genetic quadrupedal locomotion. The Ulas family of Turkey is a fascinating illustration of how a rare genetic trait can influence fundamental motor skills. These individuals present a hereditary genetic condition that leads them to walk on all 4 limbs, hands, and feet. Scientists have suggested the hypothesis of an evolutionary regression or the reactivation of an ancient genetic pattern linked to primitive locomotion. The study of this family by researchers focuses on the underlying genetics that modulate the development of the skeleton and the neuromuscular system to allow for such an adaptation. Beyond biology, their story is a powerful example of behavioral and environmental adaptation, demonstrating the malleability of the human body when faced with unusual genetic constraints. Their observations offer a unique field of study in human paleontology and developmental motor biology concerning the regulation of Hox genes and their impact on posture. This lineage raises profound questions about the plasticity of ancestral motor patterns. Number 5. The Piebald family. Familial Piebaldism condition. The Piebald family is distinguished by a genetic manifestation impacting pigmentation, familial Piebaldism, which results in irregular areas of depigmentation on the skin and hair. In this family, these unique patterns, often characterized by white streaks of hair or light skin patches, are shared by several members, testifying to a strong hereditary transmission. Piebaldism is caused by mutations affecting melanocytes during embryonic development. Research on this family allows for the refinement of the understanding of genetic factors that control the migration and survival of melanocytes in the epidermis and hair follicle. By openly celebrating these differences, the Piebald family also contributes to deconstructing social stigmas linked to pigmentary variations, highlighting the importance of accepting phenotypic diversity and facilitating the understanding of complex hereditary pigmentary anomalies. Their case is essential for modeling the impact of the MITF gene on cellular differentiation. Number 4. The Fugate family. Kentucky. Methemoglobinemia. Blue skin. The Fugate family, historically associated with the troublesome Creek region of Kentucky, is emblematic of familial methemoglobinemia, a condition that gives the skin bluish tints. This coloration is due to the presence of a rare recessive gene introduced by the ancestor Martin Fugate. Methemoglobinemia causes an abnormal oxidation of hemoglobin in the blood, reducing its ability to transport oxygen effectively, which gives the skin this cyanotic tint. Genetic studies on this isolated lineage were fundamental in identifying the precise biochemical mechanism of this mutation, often linked to a deficient enzyme. Although the condition was a source of mystery and curiosity in the past, today it serves as a model for understanding oxygen transport disorders at the molecular level, a critical field in hematology and physiology where enzymatic correction is actively researched. This work has led to the development of specific treatments for acute cases of cyanosis. Number 3. The Dilva/Doilva family. Brazil. Familial polydactyly. The Dilva family, originating from the Brasilia region of Brazil, is remarkable for the high prevalence of polydactyly, a trait characterized by the presence of extra fingers and/or toes. 14 members across several generations have extra fingers and toes that are fully functional. The fact that this condition manifests so strongly within the lineage, even when one parent is unaffected, suggests a dominant mode of genetic transmission with high penetrance. Research focuses on the genes regulating limb development, seeking to understand the molecular mechanisms that control the exact number of digital rays. The Dilva family's familial polydactyly is particularly interesting because the members have integrated their extra digits and toes as functional advantages in various activities. Their case is a reference for the study of numerical variations of human extremities and potential therapies for the regeneration of missing digits. Analysis of their extra digits reveals complete innervation and vascularization. Number 2. The Gomez family. Mexico. Generalized congenital hypertrichosis. Werewolf family. A family group in Mexico, nicknamed the werewolf family, is affected by generalized congenital hypertrichosis, CGH, causing excessive growth of thick hair over the entire body and face. This trait, which spans 5 generations with 19 affected members, is potentially linked to the reactivation of an ancient ancestral gene. Genetic studies attempt to isolate the mutation responsible for this hair proliferation, which is extremely rare, fewer than 300 documented cases worldwide. Scientific interest lies in understanding the development pathways of hair follicles and epigenetic factors that can reactivate dormant genetic sequences. The way this family has overcome social isolation to become spokespeople for genetic diversity illustrates an important shift from stigmatization to scientific and public acceptance of their morphological differences, while providing a model for deciphering the regulation of dormant gene expression. Research explores the involvement of non-coding control regions. Number 1. The Parviz-Akhtar family, UK. Panfamilial albinism. The couple Alam Parviz and Shamim Akhtar, residing in the United Kingdom, and their 6 children, present an extraordinarily rare genetic configuration. All members of the family are affected by albinism, a disorder linked to a deficit in melanin production. It is exceptionally rare for two albino parents to have offspring who all inherit this condition. This family has become a subject of genetic study to confirm the homozygous recessive transmission of the albino allele in a context where both parents carry it. Their story is also marked by a significant family reunion after a long separation, reinforcing their status as a symbol of unity. Scientifically, their case allows for the consolidation of genetic transmission models for melanin-related syndromes. Their visibility serves a larger goal by promoting the acceptance of people with albinism, who often face significant visual and social challenges linked to increased sensitivity and specific ocular deficiencies. Molecular analysis provides clear data on allelic severity. Number 11. A moment of unexpected circumstances. The Macambe family was preparing for the great harvest. Under the canopy, the patriarchs and their sons, marked by ancestral rituals, displayed their swollen ritual bellies, signs of their spiritual fertility. Their leader, his face darkened by sacred pigments, led this strange procession through the forest. Each member carried the burden of their commitment, a lineage united in a tradition that the outside world could not understand. What do you think this level of physical commitment represents for a family tradition? Is it an act of devotion or a form of coercion? These 11 family lineages illustrate the richness of human genetics, from pigment to facial structures. The professional study of these rare traits is essential for advancing medicine and our biological understanding. They open pathways toward personalized gene therapy and inspire the scientific world through their resilience in the face of adversity.